Okay, second part of the video is a bit lengthy, but lengthy probably because I have a lot of diagrams to show you, but it's actually not that much. It's just that probably your mind would get a bit, well, overblown or a bit headache because of different types of transcription factors. But let's have a look at the general transcription factors. So from the first video, we know that there are at least 6, okay, but there are a whole lot more. So general transcription factors are required for transcription in eukaryote from all genes. Well, you hate it, but still, okay, you require it, okay, for you to function. So they are also known as GTFs. So they assist RNA polymerase II in transcription initiation, which we do as much, and they are designated as such, okay, TF2 because of RNA polymerase II. So A, B up until H, and most of them are multimeric proteins, which means that they have more than one subunit. So they are equivalent GTFs, okay, which are highly conserved among the eukaryotes. So that means that, okay, different types of TF2A and B or C or D up until H, okay, in different types of eukaryotes, they are more or less have high conserved sequences. So in prokaryote, only one general transcription factor, okay, or sigma factor is required, but we know that in eukaryote there are more than 6. So let's have a look, okay, let's get to know them better. Okay, so in sequence order, okay, remember the sequence order? So TF2D, the one that has to recognize, okay, the Tata box or the promoter sequence. So it is composed of 14 subunits, one of them being the Tata box binding protein, which is the TVP over here. So the function of TF2D is to recognize the promoter and to recruit the TF2B, which comes in next. So let's have a look at the TVP or the Tata box binding protein. So this is actually how they look like, okay, but in ribbon structure, but I'm going to show you how it looks like, okay, when it has been simulated. So this is, okay, the TVP protein, okay, and this is basically how it binds, okay, to the Tata box, okay. So this is basically the nucleotide sequence. Now, okay, this part, okay, is the close-up of this, okay. So basically, okay, just for your knowledge, and if you are interested further, you can read a book, okay, about this, but I'm not going to cover further than this. So they basically, okay, are, or the nucleotides, okay, or the Tata box, okay, the sequences are basically stacked bases, and the stacking, okay, is done between, okay, phenylalanine, okay, and A base, okay, which is either one of the nucleotide bases, okay, but not between two bases, okay. So it is basically from amino acid to nucleotide, not between two nucleotides. And, okay, taking a look, okay, we have seen this just now. Okay, let's have a look, okay, at the top part, okay. This is, okay, what happens when there is, okay, a steric hindrance, okay, if A was a G, as over here, okay. So the Tata box, sometimes it could have a different variation. So what happens is that, okay, it would prefer an A. So if, okay, the A was a G, okay, so if this is an A actually, okay, but if it is a G, okay, there would be a steric hindrance, okay, which means that it will not bind as tightly, or it will recognize as good, okay, as it should be if it was an A. It will recognize it still, but not as good, okay, because by right, okay, it should be an A. So the summary of interactions, okay, between the TVP and the Tata box, okay, is as such. So we know that Tata box, okay, is making a pseudo two-fold sequence specific interaction with 2 threonines and 2 asparagines of the TVP, okay, as you can see here, THR and basically ASN. So stacking of phenylalanine, okay, against DNA bases is also shown, okay, DNA bending due to stacking, okay. Of course, it's not shown, but it is actually shown in one of the diagrams just now, okay, the second diagram. So now moving on, okay, to the next transcription factor, because we know that TF2D, okay, is the first one, okay, and it has to recruit TF2B. So TF2B is up next. So the function of TF2B is to start site selection for polymerase II. So basically, okay, it will recruit, okay, PL polymerase II, okay, to form a complex, okay, later on. So some TF2B, okay, have a mutants, and the mutants, okay, result in a shift of transcription start site, okay, which means that sometimes it is a leader start site. So under certain conditions, for example, if there is a BRI element promoter, polymerase II together with the TF2D and TF2B can form the minimal initiation complex. So at most promoters, however, usually TF2, E, F, and H, okay, are necessary, but again, in such a case, sometimes, okay, these three are not necessary. Alright, so we know that TF2B doesn't work alone. Okay, it works with the CTD or C terminal domain. So CTD of TF2B interacts with both DBP and DNA around the promoter, especially with Bri element, as you can see over here. So the rough positioning of polymerase 2 is due to the interaction of TF2B CTD with TBP. Okay, so well, your polymerase, okay, is basically not shown here, okay, but here. So fine positioning is due to interaction with DNA and N-terminal domain of the TF2B, okay, so over here, okay, interacts with the polymerase 2. Alright, so let's move on, okay, to the next one. So we have TF2F. Right, so we know that TF2F, again, what does it function? Again, what is the function? So recruitment of polymerase 2 to the existing DNA, TF2DB complex. So positioning polymerase 2, okay, over the start site and binding to the non-template DNA strand, and also sometimes reduces non-specific binding of RNA polymerase 2 to DNA. So this is the rough 3D structure of TF2F. So as you can see, so it is heterotetramer, okay, made of 2 subunits, okay, and of course, okay, sometimes, okay, the Rep2 and Rep74, okay, are dimer within the complex structure. So I'm not going to go into details. So let's move on, okay, to the other 2 transcription factors, okay, which is TF2E, okay, and H. So TF2E is heterotetrameric as well, okay, A to B2. So its function is basically, even though it appears to create a docking site, okay, it basically is the one, okay, that, well, it basically creates a docking site, okay, because without which then usually the protein-protein interaction, okay, will not occur with the RNA polymerase especially. And TF2E also modulates, okay, TF2H, we know that as much, okay, for the activities. And in addition, TF2E enhances promoter melting. Why do we need to melt the promoter? That is to start off the transcription. So the next one is TF2H. So we know that it is a multimeric protein composed of 9 subunits, blah blah blah. So the function is to have, okay, since it has a helicase activity, what is the function of helicase? Remember that back, okay? It basically unwinds the DNA duplex at the start site to allow RNA polymerase to bind to the template strand, because if it doesn't unwind, so your RNA polymerase could not start off the transcription because there is no unwinding. Okay, it's still wind together. You couldn't basically use, okay, leading strand. So TF2H, okay, also has a kinase activity, okay, which is used to phosphorylate polymerase 2 in the beginning of the elongation. And other subunits of the TF2H, okay, have been shown to recruit RNA repairing enzymes if RNA polymerase reaches damaged region in the DNA and gets stalled. So during the process, okay, sometimes it gets stalled, okay, or it gets basically backslided, okay. So they have a backup unit. Now, this is the early events, okay, in the elongation. So we know that, okay, polymerase 2 transcribes away from the start site subunit of the F2H, which phosphorylates the polymerase 2 CTD, and this would result in promoter escape. So what happens, okay, when promoter escape, okay, general factors gets released, okay, slowly. Only the RNA polymerase hangs to it, and then you can start off the transcription. Now, the last one, okay, transcription factor, I put TF2A. Now you may be wondering, why is TF2A is the last, okay, because by right, okay, it should be after 2B, right? So for transcription in vivo, okay, another transcription factor TF2A is required, okay. So it is basically have, the function is still unclear up until now, but usually it helps, okay, other factors to bind. And having said that, doesn't mean that TF2A is not important. It is important, it is needed, okay, to form the initiation complex nonetheless. And it has been shown to have some anti-repressive function as well. But TF2A is usually not required for transcription in vitro, in the lab, okay, when you basically do an enhancement of it. Alright, so moving on to other, okay, transcription factor, which is TF, okay. So basically they are TBP, okay, or Tata box, okay, binding factor associated factors, okay. So it's called TAF. So there are 13 types of TAF. Wow, apart from 6, we now have another 13, okay, to add to your misery. Okay, so some of them seem to be necessary for transcription initiation from promoters, which is lacking the Tata box. Okay, other okay, have been shown to be tissue-specific co-activators. So that means that you don't really need all 13 of it, okay, some of it is needed, okay, and hence you don't really need to know each and every one of the 13. So TAF subunits, okay, also interact with other GTFs, okay, therefore stabilizing the complex, okay, general transcription factors. So this is, okay, basically I think this almost towards the end, again, the 3D structure of polymerase 2 holoenzyme. So even though this is the polymerase 2, okay, so what happens, okay, is that, okay, this is what happens when all the transcription factors comes in place, okay. So when it has come into place, it has bind to where it's supposed to bind. You can see that, okay, this forms a holoenzyme, and it looks kind of like ugly or very complex, okay, but this is the complex that is needed to start off the transcription in eukaryote, okay. Oh, we have another one, okay, which is the TLF, okay, or TBP-like proteins, or Tata box binding like protein, okay. So in different organisms, okay, as you can see here, yeast, plants, okay, C. elegans, Drosophila, vertebrates, okay, there are different types of TLFs, and how they recognize the sequence are also different. So basically, TLF helps or kind of like act, okay, like an enhancer, okay, to the TBP. Can it take the place of TBP? Actually, not really, but in certain cases, yes, but it is not preferred. Okay, usually TBP, okay, is preferred over TLF. Alright, so TBP-like protein, okay, are TBP protein analogs, okay, not to be confused with a TAF. So some TLF recognize the Tata box, okay, and other recognize some different promoter sequences. Okay, so TLF are thought to play a role in transcription regulation as well, okay, but not to replace the TBP. So TLF as transcription regulators, okay, so this is how it works, okay. So I'm taking an example of C. elegans, which is actually the simplest to basically explain. So we know that, okay, what happens, okay, is that, okay, in C. elegans, okay, so usually you require, okay, TBP, but if TBP is not there, okay, this is where in C. elegans, okay, and again I'm saying this, okay, it doesn't happen to all eukaryotes, okay. In C. elegans, if TBP, okay, is not available, TLF is actually could be used, okay, to recognize the TLF. But, okay, if TBP is still not there, remember, okay, no transcription will occur, because, okay, TLF, okay, doesn't have the power like TBP, okay, to gather all the other transcription factors to start or initiate the transcription. Okay, again, okay, this is an example, okay, if you don't have TBP, but, okay, you have TLF and you want TLF to start off the transcription. Again, okay, it could bind to the Tata box, but it wouldn't be able to initiate transcription. So in second case, okay, you have TBP, okay, which is supposed, okay, to recognize, okay, to the Tata box, okay, but, okay, in place of TF2A and TF2B, we have TLF, okay, binding together to them, okay, not TBP over there, okay. So when these two binds together, so it means that it couldn't bind to the TBP, it couldn't go to the TBP. Again, without these two, transcription couldn't occur, okay. So again, with some other recognition sequences apart from the Tata box, okay, whereby TLF binds to it, okay, without TBP, okay, usually transcription will not occur until you find the TBP, okay. So if, okay, and there is also, okay, one unique condition, okay, whereby you have a few other sequences, okay, recognition sequences, for example, and, okay, you have your TBP already bound, okay, to the Tata box, okay, so you know that it's going to be ready for transcription initiation, but your TLF kind of like what we say in Malay is like kaca daun, okay, or basically it's playing havoc, okay, it binds to the recognition sequence upstream or downstream. So this would hinder other transcription factors from binding, and hence you still get no transcription initiation, okay. So this is how, okay, TLF, okay, sometimes could play havoc, okay, but sometimes it could also enhance the initiation or regulate the initiation. And this TLF, okay, is usually being applied, okay, in case if you want to stop a certain population from breeding, and we know that in C. elegans, okay, so this is actually how they do it, okay. So this is at 30 to 80 cell stage. At 150 to 300 cell stage, okay, TLF becomes a positive regulator, which means that it is now enhancing to speed up the process. Okay, so now, okay, but okay, TLF, okay, oh, basically it's a bit downwards. Okay, so TF, what you have here, okay, when it binds to recognition sequence, not the data box, okay, it could start off the transcription, okay, and the TBP, okay, the usual one, okay, it binds to the data box, okay, it could start off, okay, or okay, if you have such a case, okay, so this basically could now happen, but only at 150 to 300 cell stage once it has reached, okay, towards those steps only. Alright, so we have a few more slides, okay, before we break off for the second video. What is polymerase 2 backsliding, pausing, and arrest? So these are some phases that you will come across in the transcription process in eukaryote. So let's get to know them in general first. So backsliding, as the name says, you backslide, okay, kind of like when you see Michael Jackson do the moonwalk, okay, backslide, okay. So it's an event when RNA polymerase moves backwards and new made RNA gets inserted in the funnel, okay, in the basically during the unwinding. So it can be caused by incorporation of wrong NTPs, or when 3' hydroxyl, okay, loses contact with the active site, okay, of the magnesium. So if backsliding RNA piece is around 2 to 4 nucleotide long, okay, so usually this is reversible, okay, and RNA polymerase could recover itself, okay, and this is called by pausing. So they pause, they correct it, okay, and then they proceed, okay, with the transcription. But if the backsliding, okay, gets longer, 7 to 10 or more, it will get trapped, okay, in the funnel pore, which means that there is kind of like something that's stuck there, and RNA polymerase could not cover it itself. Okay, this would cause arrest. So there are consequences to arrest, whether it will stop, it will be aborted, basically stopped, okay, or basically it will just be degraded, which we'll see. I think next week, starting from next week. So arrest could be overcome by specific elongation factors which help RNA polymerase 2 to cleave the arrested RNA. So that could be done sometimes if it's not severe. And this is okay what happens when during backsliding, okay. So backsliding can be caused, okay, when 3' hydroxyl loses contact with the magnesium. So what you can see, okay, over here. So backsliding, okay, so whereby, okay, what happens, okay, is that when it should be going forward, okay, it basically goes back, okay, so it gets stuck, okay, in the funnel. So imagine that you are pouring something, okay, but it should go through the funnel, right, okay, but it goes back up, okay. So that is actually what happens with the backsliding. Just imagine that, and hopefully you could get what happened here. So movement of RNA and DNA in transcription and also backsliding, okay, so you can see, okay, it's basically here, okay, indicated by the red, okay, of the transcription and also the backsliding. So transcription should move this way, okay, this is actually the same, okay, but okay, in case of backsliding, okay, it basically moves backwards, okay. So what causes backsliding? A few causes, okay, which we will cover, okay, next week onwards. So the next one, okay, during the phase is elongation, okay. So what happens during elongation? Okay, we have elongation factors, okay, to help with this. So in vivo rates of transcription, okay, usually is around, well, 1200 to 2000, okay, nucleotides per minute, and the baseline in vitro rate is around 100 to 300 nucleotides per minute, okay. So how fast, okay, transcription rates are. So in vitro's frequent transcription pausing and arrest, okay, occur even with chromatin-free DNA, which is quite common, and large number of proteins and protein complexes exist which would regulate elongation of transcription by the elongation factors. So elongation factors, okay, usually display the following activities, okay, enabling elongation through chromatin, okay, suppress pausing because too much pausing is actually not good for the transcription, and to overcome transcription arrest, okay. So these are some of the transcription elongation factor, okay, which is S, again known as 2S. So basically, okay, well, you can see over here, basically this one, basically along the sequences, okay, and this one basically is the yellow one, okay, along sequence. So this basically how the transcription elongation factor 2S looks like, okay. So don't worry about the position, the domain, the alpha, and whatnot, okay. But okay, why do we need, okay, the transcription elongation factor? Okay, so why does it have elongated shape? The thing is, okay, TF2S, okay, gets inserted in active site of polymerase in case of backsliding, okay. And okay, what happens is that, okay, the TF2S, okay, and the polymerase 2 complex, okay, when it gets inserted, okay, so usually it will be at the active site of the polymerase 2. I actually couldn't show you here because basically if I have a 3D structure I could show you better, but this is basically 2D, so it's quite hard, okay, to show you guys, but it's basically, okay, somewhere here. All right, so domain 2, okay, will dock, okay, on the surface, okay, the domain 2 that you see just now, okay, will basically, okay, dock on the surface, and the interaction of TF2S, okay, as it happen with the arrested RNA, is basically, okay, helps to cleave the phosphodiester bond, and this is to make sure, okay, that, okay, it could be remedied, okay, so in transcription could proceed and you could stop the arrest. Okay, so the backtrack RNA fragment gets released and RNA polymerase is rescued from the arrest, so it could continue with the transcription. So elongator decondenses, okay, the chromatin, so this is what it does. And basically I will just, okay, leave you guys, okay, to this, okay, because I'm not going to go further on how it decondenses, okay, the chromatin, okay, because we know that chromatin is actually a very condensed structure. If you see the structure, it's basically kind of like wrapped in something. So when it decondenses, basically it makes sure, okay, that, well, the arrest, okay, or you could basically do the cleaving, and the cleave, okay, could be released, okay, from the whole structure, okay, from the transcription, and then, okay, well, you could proceed with the transcription process itself. All right, so that would be the end of video 2, which is very lengthy and involve lots of transcription factors, but again, don't lose hope, okay, move on to the third video.